So in our first section of Chapter 7, 7.1, we're going to be taking a look at DNA and RNA. First, we're going to look at what's called the central dogma of molecular biology. We're going to look at the discoveries that led to the knowledge of DNA structure and function. This happened in the middle of the 20th century, around the early 1950s, and it was a really, really huge discovery. And then finally, we're going to take a look at the structure of RNA and identify the three main types of RNA. So, the central dogma of biology is DNA, which has the information on how to make a cell protein. DNA is only in the nucleus and never leaves the nucleus. So the information has to get out here for how to make a protein. So DNA, the information is read and structured in a molecule called RNA. So it takes the information by a process called transcription, takes that information from the DNA, goes out to the cytoplasm where it translates that and makes a protein. We're going to see this in a lot more detail, but all you have to know for now is DNA, the information is taken by RNA, the information is taken out to the cytoplasm where a protein is made. That's called the central dogma of biology. As I mentioned, the discovery of what DNA was and what it could do was one of the most famous in all of science. And it happened in the early part of the 20th century. It happened from about 1910 to the early 50s. And one of the first experiments was an experiment done by Frederick Griffith. And in this, he took some bacteria, living bacteria that had let's call it the S strain as your diagram here. And when he injected it into mice, it caused death, and the living strain was found in the heart. But then he took another strain of bacteria, let's call that R, and it had no effect whatsoever on the mouse, and the mouse stayed healthy. He then heat treated S, this first type of bacteria, and he heat treated it, and what happened was the mouse stayed healthy. And he found that unusual. So what first killed them didn't kill them here. And then he mixed the two strains together and the mouse ended up dead. So he wondered there had to be something that was able to transfer, some molecule, he was guessing, that transferred from the one type of bacteria to the other. He didn't know what it was, but this was a very famous experiment. His results were then carried out by Oswald Avery. You'll read about that in the textbook. And make sure that you realize that Avery said, I think it's this molecule DNA that's in the nucleus. That was followed up by Alfred Hershey and Margaret Chase, Hershey and Chase, and what they did. Erwin Chargaff discovered what the letters were of DNA that had the information. We'll talk about that in a few minutes. Rosalind Franklin, a scientist that really wasn't given much credit, was able to take special type of X-rays of molecules, and those X-rays were pivotal in the discoverers, Watson and Crick, discovering what DNA was, how it was made. So let's take a look at DNA here. So before we get to the DNA, we're going to look at nitrogen bases. It turns out that the information is contained in 4 what are called nitrogen bases: adenine, guanine, cytosine, and thymine. But most of the time, they're only known by their first letter, A, G, C, and T. And it was discovered by Erwin Chargaff that G always bonds to C, and T always bonds to A. So these are the nitrogen bases. Where do they occur? Let's take a look. They occur on the inside of the ladder. The DNA molecule has a double helix shape. So imagine a ladder that's made of flexible material, and you can spin it around so it becomes twisted. That's what the DNA double helix, it's called a double helix, looks like. And they are showing you here, here's a person over here, they're showing you a staircase, and that's what the DNA helix molecule looks like. One of the things we talked about earlier was the fact that DNA has to copy itself. And DNA, when it copies itself, is called replication. So let's look up here. Here's the parent DNA molecule. Notice C always bonds to G, G, C, A, T, G, C. This is where the information is. This is where the information on how to make a protein. And a section of DNA is called a gene. A lot of times students have questions about how can just letters contain information. Well, remember that 26 letters of our alphabet create all the words, the 30 to 40,000 words in the English language? Well, we've devised systems where those letters can be represented by only 2 symbols. We've done that with Morse code, and we've done it with the computer code, 1s and 0s. Morse code was done with dots and dashes. So we can reduce information down to symbols. So here is the double-stranded DNA molecule. It unzips, an enzyme breaks it up, and then when it copies itself, a new side will come on with the nitrogen bases, and when they're done, they'll have 2 complete copies of the DNA. RNA. DNA alone can't tell your cells how to make proteins. It needs the help of RNA. So the other main player in the central dogma of biology is RNA. Remember, DNA lives in the nucleus, but the proteins are made out in the cytoplasm. So how does it get there? RNA is the answer. So let's take a look at this diagram. This diagram is comparing DNA to RNA. So our DNA, standing for deoxyribonucleic acid, is a double-stranded ladder, and here are our base pairs. Here's our A, T, C, and G. That's where the information is. There's no information on the sides of the ladder. It's just the rungs of the ladder here in the middle. And notice that T always bonds with A, C always bonds with G. Notice these colors here. And over here, we have RNA. So take a look right next to DNA. This is a double-stranded. Right next to RNA, it's only a single-stranded. And in RNA, we don't have thymine. Instead, we have uracil. So the nitrogen bases, the nucleobases, are cytosine, guanine, adenine, and uracil in RNA. So this is a really good diagram to show you the difference between DNA and RNA. And you should study this. It'll make the next few sections easier. So we have in your textbook a written comparison of DNA, RNA. Make sure you read that and look at the 3 types of RNA. There's something called messenger RNA, which copies the instructions from the DNA, carries them out into the cytoplasm. Then there's rRNA, which form the ribosomes, the organelles that are out in the cytoplasm. And then there's a very special type of RNA called transfer RNA, which brings amino acids. Proteins are made of amino acids to the ribosomes, and they're joined together to form proteins. We'll see all of that in much more detail coming up. So the lesson summary, we should know what the central dogma is. We should know the structure of DNA and RNA and the 3 types of RNA: messenger RNA, ribosomal RNA, transfer RNA, and what their role is. We'll see all of that in much more detail in the next section, which talks about specifically how we make a protein.